Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. In the quiet depths of your cells, microscopic timekeepers are ticking. With each passing second, minute, hour, and year, these biological counters gradually wind down, marking not just the passage of time, but the process we call aging. Scientists have been searching for the mechanisms behind this inevitable march toward senescence, and one discovery stands out as particularly significant: telomeres. At the ends of our chromosomes, those tightly wound packages of DNA that contain our genetic blueprint, sit protective caps called telomeres. They're often compared to the plastic tips on shoelaces, preventing our chromosomes from fraying or tangling with one another. But telomeres do something shoelace tips don't. They shorten each time a cell divides. In the early 1990s, scientists made a breakthrough. They discovered that telomeres weren't just passive structures, they were active participants in the aging process. Every time most cells in our body replicate, a small portion of these telomeric end caps gets snipped off. After enough divisions, typically 40 to 60 depending on the cell type, telomeres become critically short. At this point, cells enter a state called senescence, where they can no longer divide. This limit on cell division, named the Hayflick limit after its discoverer Leonard Hayflick, acts as a biological clock. When enough cells reach senescence, tissues and organs begin to deteriorate. Functions decline. The body starts to exhibit the hallmarks of aging, from wrinkles and gray hair to more serious conditions like heart disease, cognitive decline, and increased susceptibility to infections. But here's where the story gets interesting. Not all telomeres shrink at the same rate. Environmental factors like chronic stress, poor diet, lack of exercise, and exposure to toxins can accelerate telomere shortening. Conversely, healthy lifestyle choices appear to slow it down. This discovery revealed something profound. Aging isn't entirely predetermined by genetics. It's malleable, influenced by how we live our lives. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free and it helps us grow this community together. More intriguing still was the discovery of an enzyme called telomerase, first identified by Elizabeth Blackburn, Carroll Grider and Jack Szostak, work that would earn them the 29 Nobel Prize in Physiology or Medicine. Telomerase can actually rebuild telomeres, adding back the protective DNA sequences that were lost. The implications were enormous. If telomere shortening drives aging, could manipulating telomerase slow or even reverse this process? Had scientists stumbled upon a potential key to extending healthy human lifespan? Some organisms seem to suggest this possibility. The immortal jellyfish, Turritopsis dohrnii, can revert to an earlier stage of development when injured or stressed, essentially resetting its biological clock. Certain lobster species produce high levels of telomerase throughout their lives and show few signs of aging. They don't become weaker or less fertile with age, and theoretically could live indefinitely if not for disease or predation. But human biology is vastly more complex, and our relationship with telomeres and aging isn't straightforward. The same mechanisms that protect us from premature aging also shield us from cancer, uncontrolled cell division that occurs when cells bypass their natural limits. Our bodies maintain a delicate balance between renewal and restraint. This tension between longevity and cancer protection sits at the heart of anti-aging research. Scientists aren't just asking if we can reverse aging, but whether we should, and, if so, how to do it safely. The quest to understand and potentially manipulate telomeres represents one of the most fascinating frontiers in modern biology, where fundamental questions about life, death, and human potential converge. The story of telomeres begins in the 1930s, when two scientists working independently, Herman Muller and Barbara McClintock, observed something peculiar about chromosomes. They noticed that chromosomes had special structures at their ends that prevented them from fusing with other chromosomes. Muller named these structures telomeres, combining the Greek words telos, end, and meros, part. For decades, these chromosome caps remained mysterious. Scientists knew they were important. When telomeres were damaged, chromosomes became unstable, leading to cellular malfunction or death. But the precise nature and function of telomeres remained elusive until the 1970s, when Elizabeth Blackburn, then a postdoctoral researcher at Yale University, decided to study the chromosomes of Tetrahymena, a single-celled organism with an unusual feature. It contained thousands of mini-chromosomes. What Blackburn discovered was revolutionary. She found that telomeres consisted of a specific DNA sequence, TTGGGG, repeated multiple times. This pattern was remarkably similar across vastly different species, suggesting that telomeres were fundamental to life on Earth. Later, working with graduate student Carol Greider on Christmas Day, 1984, Blackburn discovered telomerase, the enzyme capable of rebuilding telomeres. The significance of these discoveries wasn't immediately apparent, but as researchers continued to investigate, connections between telomeres and aging began to emerge. In 1990, Calvin Harley and Carol Greider demonstrated that telomeres in human cells, grown in laboratory cultures, shortened with each cell division until reaching a critical length, at which point the cells stopped dividing. This observation aligned perfectly with the Hayflick limit, the previously observed ceiling on how many times normal human cells could divide before entering senescence. Suddenly, scientists had a molecular explanation for this limit. Telomere shortening was the clock that counted down to cellular old age. Further studies strengthened this connection. Researchers found that cells from older individuals had shorter telomeres than those from younger people. Certain premature aging disorders, like Hutchinson-Gilford progeria syndrome and Werner syndrome, were associated with accelerated telomere shortening. And some of the most long-lived species on Earth, from certain tortoises to bowhead whales that can live over 200 years, maintained their telomere length better than shorter-lived relatives. The evidence was compelling enough that in 1929, Blackburn, Greider, and Jack Szostak were awarded the Nobel Prize for their work on telomeres and telomerase. Their discoveries had opened up an entirely new perspective on cell biology and aging, but the relationship between telomeres and aging proved more complex than initially thought. While telomere shortening clearly played a role in cellular aging, it wasn't the whole story. Other factors, from mitochondrial dysfunction and protein misfolding to epigenetic changes and stem cell exhaustion, also contributed to the aging process. Telomeres were an important piece of the puzzle, but not the only one. Moreover, the very mechanism that made telomeres so intriguing as an anti-aging target also made it potentially dangerous. Cancer cells often activate telomeres to achieve immortality, dividing endlessly without hitting the Hayflick limit. This raised a critical question. Could boosting telomerase to extend lifespan inadvertently increase cancer risk? This paradox, that the same enzyme that might extend healthy life could also promote cancer, has become central to telomere research. Scientists now face the challenge of understanding how to maintain healthy telomeres without enabling malignancy. Their pursuit has led to nuanced approaches that aim to capture the benefits of telomere maintenance while avoiding its pitfalls, setting the stage for the current frontiers in anti-aging research. To understand how telomeres function as cellular timekeepers, we need to examine the basic process of cell division. When cells replicate, they must duplicate their entire genome, all 3 billion base pairs of DNA, before splitting into 2 daughter cells. This replication begins with the unzipping of the DNA double helix, followed by the assembly of new complementary strands using the originals as templates. But there's a fundamental problem with this process, known as the end replication problem. The enzymes that build new DNA strands, DNA polymerases, can only work in one direction, and they require a small RNA primer to get started. When the cell removes these primers and replaces them with DNA, it can't fill in the very end of the chromosome. With each round of replication, a small piece of DNA at the chromosome's end is lost. This is where telomeres come in. These repeating sequences of DNA, in humans the sequence TTAGGG repeated thousands of times, serve as buffers. When the end replication problem causes DNA loss, it's these non-coding telomere sequences that get shortened, not the crucial gene-containing regions of the chromosome. It's a brilliant evolutionary solution. Sacrifice disposable DNA to protect the essential genetic information. The average human telomere is about 15,000 base pairs long at birth. With each cell division, telomeres typically lose between 30 and 200 base pairs. After enough divisions, telomeres reach a critical length of about 4,000 base pairs, triggering cellular senescence, a state where the cell stops dividing but remains metabolically active. Senescent cells aren't just idle, they're actively problematic. They secrete pro-inflammatory compounds, enzymes that break down surrounding tissues, and signals that can make neighboring cells senescent too. This phenomenon, called the senescence-associated secretory phenotype, or SASP, contributes significantly to age-related tissue dysfunction and disease. Telomere shortening doesn't occur at the same rate in all cells. Highly proliferative tissues, like skin, intestinal lining, and blood cells, experience more rapid telomere erosion than tissues with lower turnover rates, like neurons in the brain. This partially explains why certain organs show signs of aging earlier than others. The telomere shortening rate is also influenced by external factors. Oxidative stress, damage caused by reactive oxygen species generated during normal metabolism, can break telomeric DNA directly, accelerating shortening beyond what occurs from the end replication problem alone. Chronic inflammation, exposure to environmental toxins, psychological stress, and poor diet can all increase oxidative stress, indirectly accelerating telomere shortening. Notably, telomere length varies significantly among individuals of the same age. Some of this variation is genetic. Telomere length is partly heritable, but lifestyle factors play a substantial role too. Studies have shown that chronic psychological stress, lack of physical activity, poor sleep quality, and unhealthy diets correlate with shorter telomeres. Conversely, regular exercise, stress management techniques like meditation, adequate sleep, and diets rich in antioxidants are associated with longer telomeres. This raises an intriguing possibility. Could changes in lifestyle slow the rate of telomere shortening, potentially extending healthy lifespan? Evidence suggests yes, though the effects aren't dramatic enough to truly reverse aging. A 2013 study found that men who adopted comprehensive lifestyle changes, including a plant-based diet, moderate exercise, stress management, through yoga and meditation, and increased social support, showed a significant increase in telomerase activity and a modest increase in telomere length over a 5-year period. But the most powerful intervention would be directly activating telomerase to rebuild shortened telomeres. This is where the enzyme telomerase becomes crucial. While most somatic body cells produce little or no telomerase after early development, certain cells, namely stem cells, germ cells, and, unfortunately, cancer cells, maintain telomerase activity throughout life. Understanding how telomerase is regulated in these cells has become a central focus of anti-aging research, as scientists seek ways to selectively activate this enzyme in normal cells without triggering cancer development. The frontier of telomere research spans multiple approaches, from pharmacological interventions to genetic engineering and regenerative medicine. Scientists are pursuing various strategies to maintain or restore telomere length safely, each with its own set of challenges and potential. One promising avenue involves small molecule compounds that can activate telomerase. The most studied of these is a plant-derived compound called cycloastragenol, marketed as TA65. This telomerase activator has been shown to lengthen telomeres in certain human cells in laboratory settings. Initial human studies have yielded some promising results, improvements in immune function markers, and modest lengthening of telomeres in some participants. However, the effects are relatively small and long-term safety data remains limited. Another approach focuses on delivering the gene for telomerase, TERT, directly to cells. Several research teams have developed gene therapy techniques to introduce TERT into adult animals. In a landmark 2012 study, researchers at the Spanish National Cancer Research Center used viral vectors to deliver TERT to adult mice. The treated mice showed significant improvements: extended lifespan, reduced age-related diseases, and improved markers of metabolic health, all without increased cancer rates. This suggested that carefully timed and targeted telomerase activation might be safe and effective. Scientists are also exploring indirect methods of telomerase activation. Certain supplements like resveratrol, found in red wine, and N-acetylcarnosine may support telomere maintenance through their antioxidant properties, reducing the oxidative damage that accelerates telomere shortening. However, the evidence for these approaches remains preliminary, with relatively modest effects compared to direct telomerase activation. Beyond telomerase, researchers are investigating the body's natural mechanisms for dealing with critically short telomeres. When telomeres become too short, they trigger a DNA damage response that leads to senescence or cell death. Understanding this response pathway could reveal new targets for intervention, perhaps not preventing telomere shortening itself, but modifying how cells respond to it. The emerging field of xenotherapeutics offers a complementary approach. Rather than preventing senescence by maintaining telomeres, xenotherapeutics aims to selectively remove senescent cells, senolytics, or neutralize their harmful secretions, xenomorphics. Several senolytic compounds have shown remarkable effects in animal models, rejuvenating various tissues and extending healthy lifespan. The first human trials of senolytics are currently underway, with preliminary results suggesting potential benefits for age-related conditions like idiopathic pulmonary fibrosis and diabetic kidney disease. Another frontier lies in the relationship between telomeres and mitochondria, the cell's power plants. Recent research has revealed that telomere shortening affects mitochondrial function and vice versa. This mitochondrial telomere axis appears crucial to the aging process, with dysfunction in either system accelerating problems in the other. Interventions targeting both systems simultaneously might prove more effective than focusing on telomeres alone. The study of telomeres has also expanded beyond their physical length to include their structural integrity and protein composition. The shelterin complex, a group of 6 proteins that protect telomeres, plays a vital role in telomere maintenance. Mutations in shelterin components can lead to premature aging syndromes, suggesting that telomere function, not just length, is critical for longevity. Perhaps the most ambitious frontier is the development of techniques to measure biological age more precisely. Telomere length is just one biomarker of aging. Others include epigenetic changes, protein glycation, and inflammatory markers. By integrating these into comprehensive aging clocks, researchers hope to better assess the effectiveness of anti-aging interventions and tailor treatments to individual needs. As these research frontiers advance, they're yielding insights that extend far beyond simple telomere manipulation. They're revealing aging as a multifaceted process, one where telomeres play a crucial but not exclusive role, and where effective interventions will likely require sophisticated, multi-targeted approaches. The relationship between telomerase, and cancer represents one of the most significant challenges in anti-aging research. It's a paradox that sits at the heart of human biology. The very mechanism that might extend our lifespan also serves as a key enabler of our most feared diseases. Cancer develops when cells escape the normal controls on division and growth. One of the body's primary defenses against this uncontrolled proliferation is the limit on cell divisions imposed by telomere shortening. When telomeres reach a critical length, cells typically enter senescence or undergo programmed cell death, apoptosis, preventing them from dividing indefinitely. This constraint serves as a crucial anti-cancer mechanism, a biological barrier that potential cancer cells must overcome to become truly malignant. Indeed, approximately 90% of human cancers reactivate telomerase to maintain their telomeres, granting them a kind of cellular immortality. Most accomplish this by mutations that increase expression of the TERT gene, which codes for the catalytic component of telomerase. The remaining 10% use an alternative mechanism called ALT, alternative lengthening of telomeres, which maintains telomeres through DNA recombination rather than telomerase activity. This widespread telomerase activation in cancer cells presents a conundrum for anti-aging researchers. If we boost telomerase activity to extend healthy lifespan, might we inadvertently increase cancer risk? Conversely, if we inhibit telomerase to fight cancer, might we accelerate aging? The answer appears to depend on context and timing. In 2010, researchers at Harvard Medical School demonstrated this complexity in a remarkable experiment with mice. They engineered mice lacking telomerase, which aged prematurely due to rapid telomere shortening. When they reactivated telomerase in these prematurely aged mice, the animals showed dramatic reversal of age-related damage, improved fertility, neural function, and intestinal health without developing cancer. However, the same intervention in young non-prematurely aged mice did increase cancer rates. This suggests that the consequences of telomerase activation depend critically on the existing state of the cells and tissues, beneficial in already damaged tissues with short telomeres, but potentially harmful in younger tissues where precancerous mutations might be present. The timing of telomerase activation also appears crucial. Long periods of telomerase expression may increase cancer risk, while intermittent, carefully timed activation might provide anti-aging benefits without significantly elevating cancer risk. This has led to the concept of telomerase pulse therapy, brief periods of telomerase activation followed by periods without activation, potentially capturing the benefits while minimizing risks. Another promising approach involves enhancing the body's natural cancer-fighting capabilities alongside any telomerase-activating intervention. For instance, boosting the immune system's ability to detect and eliminate potentially cancerous cells might counterbalance any increased cancer risk from telomerase activation. Scientists are also developing more targeted approaches to telomerase activation. One strategy involves selective delivery of telomerase to specific cell types most affected by aging but least prone to cancer development. Another focuses on activating telomerase only in cells with critically short telomeres, where the benefits would outweigh potential risks. Perhaps most intriguing is research suggesting that moderate telomerase activation might actually reduce cancer risk in certain contexts. Very short telomeres can lead to chromosomal instability, a hallmark of many cancers. By preventing telomeres from reaching this critically short length, carefully calibrated telomerase activation might maintain genomic stability and reduce cancer-causing mutations. The cancer paradox remains one of the most significant challenges in anti-aging research. Resolving it will require sophisticated approaches that can distinguish between healthy and potentially malignant cells, activate telomerase in a controlled manner, and simultaneously bolster natural anti-cancer defenses. Success would represent not just a victory against aging, but a deeper understanding of the fundamental trade-offs that evolution has built into our biology. While telomeres represent a crucial piece of the aging puzzle, they're just one aspect of a much larger picture. Modern aging research has identified at least 9 distinct hallmarks of aging, biological processes that collectively define age-related decline. To truly understand the prospects for anti-aging interventions, we need to explore how telomeres interact with these other hallmarks and consider approaches that address multiple aspects of aging simultaneously. Beyond telomere attrition, the hallmarks of aging include genomic instability, epigenetic alterations, loss of proteostasis, protein maintenance, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. Each of these processes contributes to aging in distinct yet interrelated ways. Importantly, telomere shortening doesn't occur in isolation. It both affects and is affected by these other aging processes. For instance, oxidative stress from mitochondrial dysfunction can accelerate telomere shortening. Conversely, telomere shortening can trigger cellular senescence, which in turn promotes inflammation and tissue dysfunction through the senescence-associated secretory phenotype, SASP. This interconnectedness suggests that truly effective anti-aging strategies will need to target multiple hallmarks simultaneously. Several approaches show particular promise in this regard. Caloric restriction, reducing calorie intake without malnutrition, appears to address several hallmarks at once. It improves mitochondrial function, enhances proteostasis, modulates nutrient-sensing pathways like insulin, IGF-1, and mTOR, and may even slow telomere shortening. Various forms of intermittent fasting appear to capture many of these benefits, while being more sustainable than continuous caloric restriction. Exercise similarly affects multiple aging pathways. Regular physical activity improves mitochondrial function, reduces inflammation, enhances proteostasis, and may even boost telomerase activity in certain tissues. A 2018 study found that highly active adults had significantly longer telomeres than sedentary individuals, corresponding to approximately 9 years of reduced cellular aging. Several pharmacological approaches also target multiple aging hallmarks. Rapamycin, which inhibits the mTOR pathway, extends lifespan in various organisms from yeast to mice. It improves protein quality control, enhances mitochondrial function, and may indirectly support telomere maintenance. Metformin, a diabetes medication, similarly affects multiple aging pathways and is currently being studied in the TAME, Targeting Aging with Metformin, trial, the first major clinical trial specifically designed to test an anti-aging intervention in humans. Senolytics, compounds that selectively eliminate senescent cells, represent another promising multi-target approach. By removing these dysfunctional cells, senolytics may mitigate the harmful effects of cellular senescence, while potentially reducing the burden on telomere maintenance systems. Early animal studies show remarkable effects, including extended healthy lifespan and improved function across multiple organ systems. The emerging field of epigenetic reprogramming offers perhaps the most comprehensive approach. In 2016, researchers at the Salk Institute demonstrated that partial reprogramming of cells using Yamanaka factors, 4 genes that can reset cell identity, reversed multiple markers of aging in mice, including some aspects of telomere biology. This approach essentially resets the epigenetic landscape of cells without changing their identity, potentially addressing many hallmarks of aging simultaneously. These holistic approaches reflect a growing recognition that aging is a system-wide process. While telomere maintenance remains an important target, intervening at multiple points in the aging network may yield more substantial and sustainable benefits than focusing on telomeres alone. This broader perspective also highlights the importance of personalized approaches to anti-aging. Individuals age differently. Some may experience more rapid telomere shortening, while others might show more pronounced mitochondrial dysfunction or proteostasis decline. Comprehensive assessments of biological age across multiple parameters could enable tailored interventions that address each person's unique aging profile. As research progresses, the most promising anti-aging strategies will likely combine targeted approaches like telomerase activation with broader interventions that address multiple aging hallmarks, all personalized to individual needs and risks. This comprehensive approach recognizes aging not as a single process to be reversed, but as a complex system to be rebalanced. As science advances toward potential interventions that could significantly extend human lifespan, we face profound ethical questions that extend far beyond laboratory techniques and clinical trials. These questions touch the core of what it means to be human, how we structure our societies, and what responsibilities we bear toward future generations. Perhaps the most fundamental question is whether radically extended lifespan is desirable at all. Some philosophers and bioethicists argue that mortality gives meaning to human life, that the finite nature of our existence creates urgency, preciousness, and significance. Others counter that involuntary death from aging represents a tragedy to be overcome, and that longer lives would allow for greater human flourishing, deeper relationships, and more meaningful projects. The potential social implications of anti-aging technologies raise equally complex questions. If effective treatments emerge, who would have access to them? In our current healthcare systems, would these interventions be available only to the wealthy, potentially creating a two-tiered society where some age and others don't? Such disparity could exacerbate existing inequalities and create new forms of discrimination based on biological rather than chronological age. There are also concerns about environmental sustainability. Earth's resources are already strained by our current population. If people began living significantly longer lives, how would that affect resource consumption, population dynamics, and intergenerational equity? Would longer lives lead to career stagnation with fewer opportunities for younger generations? Or would extended health spans create new possibilities for productivity and creativity throughout longer careers? The answers aren't straightforward. Extended healthy lifespan could actually reduce healthcare costs by compressing the period of late-life disability. People might have fewer children if they had longer reproductive time spans in which to start families. And societies might adapt with new educational models, career structures, and social institutions designed for longer lives. Religious and cultural perspectives add further dimensions to this ethical landscape. Many religious traditions incorporate specific understandings of mortality, natural limits, and divine purposes that may conflict with radical life extension. Different cultures value youth, elderhood, and the life course in vastly different ways. As anti-aging technologies advance, these diverse worldviews will shape both individual choices and policy decisions. Scientific uncertainties complicate these ethical considerations. We don't yet know whether telomere-based or other anti-aging interventions would truly extend healthy lifespan or merely prolong the period of late-life disability. We don't know whether such treatments would affect different organs and systems uniformly, or whether they might preserve physical health, while cognitive abilities decline, or vice versa. These uncertainties make it difficult to assess potential benefits and harms. There's also the question of how far we should go in manipulating fundamental biological processes. Is aging a disease to be cured, or a natural process to be respected? Are there meaningful distinctions between treating age-related diseases individually and targeting the aging process itself? Different stakeholders, from scientists and physicians to religious leaders and policymakers, may draw these lines differently. Despite these complexities, there are some ethical principles that could guide the development of anti-aging technologies. Transparency in research, equitable access to resulting treatments, careful long-term safety monitoring, and respect for individual autonomy in choosing whether to use such interventions. These principles could help ensure that advances in longevity science benefit humanity broadly, rather than exacerbating existing inequalities. As research on telomeres and other aspects of aging advances, these ethical questions will become increasingly urgent. The decisions we make now about research priorities, regulatory frameworks, healthcare systems, and social policies will shape how anti-aging technologies develop and who benefits from them. This makes it essential to include diverse perspectives in these conversations, engaging not just scientists and policymakers, but philosophers, religious leaders, social scientists, and ordinary citizens from across cultures and generations. The quest to understand and potentially modify human aging through telomere science represents one of the most profound scientific adventures of our time. Whether it leads to modest extensions of healthy lifespan or more radical transformations of the human condition, it will surely change how we think about our lives, our societies, and our future. The ethical questions it raises may ultimately prove as challenging and as important as the scientific questions themselves.